Remember, surface area to volume ratio needs to be high so that cells can diffuse nutrients and wastes efficiently. If they start getting too big, the ratio becomes compromised, and that's why cells can't grow indefinitely. They need to divide. They're also going to need to be replaced if there's any damage that happens. So for example, if you skin your knee, you're going to want to replace those skin cells pretty quickly so that you're not going to go ahead and compromise innate immunity. And cells are going to need to be replicated a ton during any type of embryonic growth or development. And then all of these are forms of asexual reproduction so that you can go ahead and perform mitosis to get those cells to divide. So the cell cycle is going to consist of two major phases. One of them is going to be interphase, which you see right here, and about 90% of the cell cycle is going to be spent in that phase. And then the other 10% is going to be in this mitotic phase. This mitosis is going to be a completely different video for later. We're going to focus on this idea of interphase right now. So if you break up interphase, again, there's three different sections. So in G1, which is what this is, you're going to go ahead and have the cell grow, it's going to enlarge, it's going to make RNA, and it's going to make proteins. In the S phase, which this is, the synthesis phase, that's where the DNA is copied and the chromosomes are replicated. And that's also where these special organelles called the centrioles, they're going to replicate too. And then there is G2, and that's going to be where the cell is still making RNA and proteins, but now it's preparing to divide. There's also something called a G0 phase. So some cells never leave G1, okay? Well, actually, if you think about it, they're never entering S. So if they never leave G1, they kind of go into this G0 phase. They're never dividing. Basically, they're arresting the cell cycle. So animal examples of cells that do this are nerve cells, muscle cells, and technically red blood cells. So the speed at which our cells go through the cell cycle depends on types of our cells. For example, if you have skin, bone marrow, lining of the intestines, these guys are going to divide really rapidly, and they're going to go ahead and continually pass through the cell cycle phases, and they're going to replicate. But like we said before, there are other cells like brain cells or neural cells that are going to rarely divide, if ever, and that's where they stay in G0. So control of the cell cycle is really complex, but simply put, it basically deals with trigger proteins in the cytoplasm that allow the cell to go from one phase to another. Cells will accumulate those proteins during different phases, and when there's a sufficient amount present, the cell is going to go ahead and go to the next cell cycle phase. An example of these are going to be cyclins. These are a family of proteins involved in the progression of cells through the cell cycle. And they are going to be basically regulatory subunits of the protein kinases. And those are going to be the enzymes that add phosphate groups to other molecules. And again, those are going to help control cell cycle events. So a cyclin is going to form a complex with its partner. That's a cyclin-dependent kinase, okay, a CDK. And that's going to go ahead, and the cyclin with the CDK, those are going to go ahead and activate the protein kinase function. So cyclins are going to go ahead and help regulate, based on their accumulation, one phase to the other. So cyclins are named because their concentration varies in a cyclical fashion during the different phases of the cell cycle. They're going to be produced or degraded as needed in order to drive the cell through those different phases. So factors like size, again, the phase, hormones, position in the tissue, all of those can influence the accumulation of those proteins. So if we look at this, we can look at G1, okay? So here's going to be our G1 phase. So we're going to be looking at two different types of cyclins. So cyclin D and E are going to go ahead and bind to different, you can see that happening here too, cyclin D in this case. They're going to bind to their very specific CDKs 4 and 6, and they're going to go ahead and accumulate, right? They're going to accumulate. And they're going to accumulate to the point where they signal the cell to prepare the chromosomes for replication. And that means that the cell can go ahead and move. When they hit a certain type of concentration, they can actually go ahead and trigger and move to the next phase, which happens to be the S phase. And that's where the cell can replicate its DNA. And those specific cyclins in the G1, they're going to go ahead and detach from their CDK. That's going to inhibit the enzyme's activity. And then a new group of cyclins and CDKs associated with that particular phase are going to start to build up and accumulate. And again, that will eventually, at certain levels, trigger the next cell cycle phase. Regulatory features like cyclins and CDKs are really important in controlling the phase that the cell cycle is in, and it also helps control the pace. So these play a role in our checkpoints, and there are 3 of them that you have to know. So in terms of the first checkpoint, that is going to be G1. And in G1, we're going to check basically for the cell size. Did it grow enough? Are there enough nutrients? Is there any DNA that's damaged? If the answer is everything's okay, well, then the checkpoint is reached and you go to the next phase. If something is wrong, the cell can arrest the cell in G1 and try to go ahead and fix that. If it goes to the S phase, again, your DNA is going to go ahead and replicate. You're going to have double the amount of information, and then you're going to go to G2. And G2 is basically a checkpoint that really just ends up looking for cell size and DNA replication. And basically what that means is the cell is just preparing to go into mitosis. And then you've got in mitosis, the last checkpoint you have to know about, where it's checking for the spindle fiber assembly. So is the chromosome attachment correct? Is it attached to a spindle fiber? Is everything okay to rip the chromosomes apart so that you can have good and clean division? So a proto-oncogene is going to be a gene that is going to go ahead and stimulate normal cell growth and division. So for example, you'll have a normal growth factor binding to what looks like a tyrosine kinase receptor. You're going to have normal signaling cascades, signal transduction is going to take place, and then you're going to have, again, a normal transcription factor that is going to go ahead and promote division. In some cases, those proto-oncogenes mutate and they turn into oncogenes. So the proto-oncogenes are the good guys, and the oncogenes, those are the bad guys. Those are the ones where the proto-oncogenes mutate and become cancerous. So a proto-oncogene might cause a normal growth factor to change when it mutates. And that mutation is going to occur on the DNA where the cell cycle proteins are coded for. And then these regulatory proto-oncogene proteins become oncogenes. That could lead to something like an overstimulation of certain growth factors. So in this case, you have this growth factor, and then RAS is an oncogene that's going to go ahead and promote the cell to go ahead and divide unchecked. So again, here you've got this normal growth factor. The cell is growing under normal conditions. It's going to replicate, and that is the proto-oncogene. If that mutates, then we can lead to cell division that is unregulated. So here is an example of a proto-oncogene, so a master regulatory gene that is known as a tumor suppressor P53. So what this does is this is an enzyme that checks for damage. If the DNA somehow is damaged, the P53 is going to go ahead and do one of two things. It can go ahead and trigger DNA repair enzymes to go ahead and get to work. And if that doesn't work and you can't fix the cell will go ahead and start deploying apoptotic pathways. However, if this P53 mutates, it can become an oncogene and no longer function. That doesn't mean you're going to get cancer. That just means that the protein that checks for the damage isn't working. So as long as the cell cycle checkpoints and other proteins function and replication goes well, everything's fine. However, if there is damage in replication, the cell might not recognize it. And if the cell continues to divide, that's where it continues to build up potentially mutations that aren't stopped and aren't repaired. And then eventually, if that continues to happen, you can lead to the development of cancer. And what that is uncontrolled cell growth. So when you lose this control of the cell cycle and you have cancer, obviously the growth factors and signal transduction pathways are completely compromised. So one thing that ends up happening is the cells will go ahead and replicate limitlessly. They will continue to replicate. Those tissues will start to create a ton of blood vessels, and they will start to feed themselves more. They're going to evade apoptosis. Apoptosis. Those cells are going to go ahead and completely start going and overgrowing, and those cell growth signals are going to start to overexpress, and then the cells are going to go ahead and replicate faster. Okay, and they're not going to listen to any of the checkpoints any longer. The tissues can also go ahead and start to invade other tissues. So normally what's supposed to happen is a kidney cell is supposed to go ahead and only grow on another kidney cell. But if there are mutations in anchorage dependency, then what can end up happening is that cell can break off from the kidney and then travel in the bloodstream or the lymph and land on another type of cell and grow there. And the other thing is most cells in the body are density dependent. So once they get to a certain density, they stop growing. Cancer cells, again, because they don't care about growth signals, are going to go ahead and grow with unlimited growth. So that's the cell cycle. Hope that's helpful.